The little red light's on. That means it's on, doesn't it?
Okay, so our next speaker is Barbara Holland, and she's going to tell us about modelling heterogeneity in multi-gene datasets. Thanks, Barbara.
Yeah, so I noticed that a lot of the people at this conference were people who come to the New Zealand sort of phylogenetics meetings, that they're probably bored by hearing me talk about networks, and so I thought I'll actually talk about one of my PhD students' work instead. Also motivated by the fact that I got more questions about his work at the Evolution conference than about my own, which was a bit distressing. So anyway, I've decided he must be doing interesting stuff, and I've stolen it to talk about. But this work's mostly by Klaus Schliep, who's a PhD student of mine and Mike and David's. And sort of looking at this quite, I guess, current question of how should you best model multi-gene data sets. So I guess when molecular phylogenetics kicked off, it was sort of a luxury to have an alignment on a single gene. But nowadays we're more often in the situation where we might have 3 or 5 or even 100 genes on the same set of species. And those sorts of data sets create some interesting challenges because, well, I mean, one thing people will probably talk about is this sort of the gene tree, species tree incongruence problem. So your gene tree might not be the same as each other, might not be the same as the species tree. Also, genes have different functions, so it's likely that they're going to be evolving under at least slightly or perhaps very different processes. So what's the best way to model the fact that you've got these large alignments, but different parts of that alignment might be evolving under very different processes? So it's probably not the best idea just to concatenate everything and treat it like it's come from just one process. So the example data set that Klaus loves to play with is this one from the Rockes et al. Paper, which I think lots of people have picked on as a nice data set to play with. It's 106 genes from 8 different yeast taxa, and this is just here we go. This is just the consensus network of those 106 trees. You can see there's a lot of agreement between them, but there's a few places where different gene trees are sort of finding different splits. And this little movie here is just showing sort of the different edge lengths you get some of those gene trees. You can see those genes aren't all doing exactly the same thing. In fact, some of them are quite weird compared to the others. So how should we try and sort of model a data set like this? So, I mean, 2 extreme approaches you could consider would be just to say, do a concatenation type approach and say, I'm just going to fit one tree to this big concatenated data set. So that would give us, so if we're just considering branch lengths and is our sort of parameters here. So we've got 8 taxa, so we've got 13 edges in our tree. So we could say, okay, we're just going to fit this data onto a single tree. So we've got 13 parameters for edges. The other extreme would be to say, well, we want to allow each gene to have its own branch lengths. So then we could fit a model with, say, this perhaps absurdly large number of parameters. Okay, so it seems like neither of these things are probably a very good idea. If we have too few parameters, we might be introducing some kind of model misspecification and biasing our phylogenetic estimate. If we have too many parameters, it's going to sort of cause problems as well. So is there some sort of happy medium? So Klaus has come up with 2 different ideas to try and sort of solve this problem. And the first one of them is kind of based on the k-means algorithm that gets used a lot in statistics, where he says, okay, so one idea would be to have classes of genes that have the same set of edge lengths. So, but how do we sort of allocate genes to these classes? So basically, the way it works is it just starts off with each gene gets randomly put into a class, and then you start sort of moving things around. And then so your number of parameters here, so we're still thinking of this Rockes data set, so you'd have one set of edge lengths for each class. And at the moment, I mean, you could, of course, allow other model parameters to vary between classes, like the sort of specific, say, GTR model you're using or your rates across sites. But for the moment, we've just sort of considered the special case where most of the model is fixed, but we're just letting the branch lengths sort of be different between different classes. So start off, just randomly assign each of your N genes into one of the K classes, and then do your sort of standard maximum likelihood approach within that class. So concatenate the genes that have been allocated to that class, just do a sort of a single standard maximum likelihood approach. So then once you've got your sort of optimized parameters for each class, you can go back and look at each gene and say, well, what class do you get the best posterior probability in? So for each gene, you don't just look at the class it's been allocated to, you look across all of the classes and decide where does it actually fit best. And then step 4 is basically to do a big reshuffle. So you move each gene into the class where it gets highest posterior probability. And Klaus played around with sort of different things that you could do here. So in his algorithm, basically everything moves at once at that step. You could also consider just allowing one gene to change its class at a time. It didn't have such good sort of convergence. So you go around this loop until you reach a point where no genes sort of want to change class. And I mean, this is heuristic. If you run it from a different random starting sort of partition, then you can end up at a different solution. So this is an example with that Rockus data set. So we've got 106 genes here, all sort of represented by these dots. And we've said, OK, try and put these into 4 different classes. And when we start off, we've just randomly allocated each gene to one of these 4 classes and gone away and optimized our parameters for each class. Actually, they start off by looking sort of quite homogeneous, quite similar. So the next step, things are allowed to move into a class where they have this better posterior probability. And even after a single iteration, you can already see sort of in this principal components analysis that you're starting to get separation of this sort of red class. The other ones are still a bit sort of muddled up. As we step through, you start getting these gene trees. They're allowed to have quite different properties. So these ones that are in the red class, they've got a very long edge to the outgroup Candida albicans compared to some of these other ones. And so typically this algorithm only takes less than 10 sort of iterations to converge to something. So there, I think that's the final one. So this has taken sort of 9 iterations. And like I said, if you run it from a different sort of random start, you might get to a slightly different solution. So it's not sort of a definitive way of finding these partitions. So I guess the obvious question is, well, why did you choose, 4 classes? Why not some other number? And so you can try and sort of answer that question. Obviously, if you have more classes, so 1 here is just like this one extreme where you're just allowing one set of branch lengths, and 106 is the other sort of extreme where you're allowing every gene to have its own set of branch lengths. And in the middle here, we have, say, 4, 6, 8, 10, 12 different classes. And your log likelihood is basically getting better as you allow more of these partitions. See, there's a little dip here. That's just an artifact of this getting stuck in local optima when you're doing your sort of random starts. But if you've done enough sort of different random starts and taken the best, that you'd expect this to be sort of monotonically increasing. But you can also look at the Akiki information criterion and try and come up with an optimal number of classes. So for this Rockus et al. Data set, 10 partitions sort of seems to work best according to this AIC criterion. So that could be one way of trying to get around this problem of sort of too few parameters or too many. And then another interesting thing Klaus has just started to have a peek at is, so you end up with these genes, and they've been put in different classes with, do these actually match anything we know about function? So I went away and looked at the sort of Go ontology, gene ontology or Go term for a bunch of these different genes. Said, well, does that match up with the classes found by the stochastic partitioning algorithm? And at first sort of glance, found that actually seems to be it's not an exact match, but it's quite a good match. So this algorithm is actually clustering genes together that do really have similar functions. So that's, an interesting thing in itself, that you might be able to sort of turn this around and use it as a way to actually classify unknown genes as to what their function is. Okay, so that was one idea. So the second idea that is looked at is basically penalised maximum likelihood. So that's been applied in phylogenetics before by, I think, Sanderson and sort of the rates algorithm for rate smoothing. But this is the same idea but slightly different in how it's been applied. So in this case, we're going to go back to letting each gene have its own branch lengths or its own sort of set of model parameters, but add a penalty term if those parameters get too different between different genes. So, so you have basically your sort of a standard sort of likelihood. You're just sort of summing up your likelihood over, in this case, say for the Rockers data set, 106 genes. But you've also got some penalty term where, so lambda is some sort of flexible, so you choose lambda, that's how much you're penalising when things are different. So if you set your lambda equal to 0, then there's no penalty whatsoever. So this will just disappear and you're back to the situation of allowing each gene to just have its own set of parameters. Whereas if you set that lambda very high, then there's a strong penalty for having your edge lengths different between different trees. So now this is some magic that Klaus has found that I don't fully understand, but if you have questions about it, I can email him and he will presumably get back to me by the end of the week. But so okay, so how does this penalty, so we like to kind of think about parameters in terms of degrees of freedom. But what we've got is this lambda term, our penalty term, that could range between, say, 0 and infinity. So what he's using is this result by Massey and, how do you say that, Tibshirani, which basically allows you to sort of turn that parameter you're choosing your penalty term and sort of think of that in terms of degrees of freedom. So that turns out to be quite useful, because if we can actually sort of convert that in a sense into degrees of freedom, then we can use these nice things like the AIC and the BIC that will allow us to choose, you know, what is actually the best setting for this parameter here. So instead of having to guess at what a good penalty term could be, we can actually use this formula, can think of it in terms of degrees of freedom, and then let the AIC or the BIC choose what a good value for that parameter is. So again, with our yeast example, so here he's just picked out 4 out of those 106 genes. And we're starting off here allowing the branch lengths to be, so we've got our lambda, our penalty term, but we've converted that to be in terms of degrees of freedom. So up here we've got basically no penalty term whatsoever. We've got 52 degrees of freedom. So again, we're just thinking about the edge length. So that 52 is 4 times 13. Each thing is allowed to have its own set of edge lengths, sort of completely independent of what the other gene is doing. But then as we increase our sort of penalty term, we are effectively reducing our degrees of freedom and forcing these trees to become more alike. So you can see as we're clicking through here, degrees of freedom is reducing and our edge lengths are getting more similar. And we can use sort of the AIC criterion or perhaps the BIC criterion to choose what's a sort of an optimal way to do that. So that's according to the AIC, this is our sort of optimal number of degrees of freedom, whereas according to a BIC, it's less. So these are, I guess, so two ideas that Klaus has sort of been playing with to try and get around this problem of how many parameters do we really need to model these kind of data sets. I did have, one last slide here before I get to the end. I think I've got a reasonable amount of time. So you can also do interesting things like track how particular edges change as you let that degrees of freedom drop. So you can see this edge here, the red edge, that's very different in one of those 4 genes, but quite similar in the other 3. Whereas if we pick on a different edge, like the blue edge shown down here, if it's allowed to be, that edge is actually very different in all 4 trees. So you could imagine some approach that said, well, why not force, say, these three actually to be equal? Like, maybe there's some other sort of variable selection you could use that would be more sort of sophisticated. But, okay, so the real take-home message of my talk is that it's actually been very interesting supervising someone who comes from a statistics background. And Dave Penny, my boss, always makes jokes about tame mathematicians and how it's very good to try and find a tame mathematician, though he doesn't consider many of the mathematicians he's met to actually be tame. But even if you can find a wild one, that's considered quite good. But yeah, it seems to me that tame statisticians are very useful too, and they've been thinking for a long time, obviously, about these problems of how many variables do you really need to model something. And some of the tools that are sort of already out there can be very usefully applied to phylogenetics. So, thank you very much. Yeah.
In the first approach you presented to allocate genes to different classes, you mentioned that at each step you would need to optimize parameters.
Yeah.
I was wondering, wouldn't these be very computationally expensive to optimize parameters at each step?
At each step, yeah. You could, yes, I mean, is the short answer, it would be. I mean, that process is taking, say, 8 or 9 iterations to get to a conclusion, and you're having to re-optimize those parameters each time. So yes, it is slow. I mean, you could certainly, I guess, try and think of shortcuts that where you wouldn't be doing, say, a full optimization each time, or if you used where you got to last time as a starting point, perhaps you'd be able to make some shortcuts. But yes, that is probably slower.
And does it have any relevance? Because still you wouldn't be able to guarantee you are reaching a global optimum. Does it have any relevance for your approach, or not really? You can reach a local optimum and it will be.
Yeah, so he found, that running it from different random starts, you were getting sort of subtly different local optima. Not very often, I think, I mean, you'd have to do a lot more exploring to sort of say how much of a problem that really was. But I guess, yeah, and that, I mean, if you want to do different random starts, that's another sort of computational overhead, in that you're having to do everything a number of times, which is slowing it down even more.
Is there a mixture model behind the algorithm? I mean, can you write down a posterior distribution that the whole thing is, the equivalent of the least squares criterion for?
I think they're definitely, I think it is basically a mixture model underlying. Although, yeah.
Can you write that down? Does it exist, or is it in some sense the model changing as we go along?
Okay, I'm going to have to do the email option on that question.
Yeah.
Yeah.